Well, now we are turning out over to English. First of all, I would like to give a very warm welcome to Johan Emilsson from the Spring Bioscience. Sorry, I think it's the first time you're going to perform for a Danish audience. Is that correct?
Well, it's actually at least it's the first time in Jutland.
I'm very happy that you're here. I have understood that you're working in cancer, different cancer types, but I think you will be able to tell us much more about that. So the stage is yours.
Thank you very much. Perfect. All right. So really happy to be here. I spent most of my career, or my academic career, in Denmark. So as alluded to before, I'm happy to take some questions in Danish if you so want afterwards. I'm going to present Sprint Bioscience. I'm also going to present a company that might have slightly a different business model compared to other biotechs you've listened to before. So quickly, Sprint Bioscience, briefly. The purpose of the company is to extend and improve the life of cancer patients, but also being able to do so as a sustainably profitable growth company. So the purpose of the company we built over the years is that we're here for the long run. We're building a company where we are aiming towards being a sustainably profitable company. And we're getting there. We've been around for a while. We've been around now for close to a little bit more than 16 years. And we are a company that focuses exclusively on drug discovery and early preclinical work. So we do only the kind of the innovation part of finding new first-in-class targets where we can make very highly specific and highly potent small molecules to inhibit those proteins. The core of what we do is immune oncology. We also work with tumor metabolism and also how to modulate the tumor microenvironment. So essentially what happens around the primary tumor and how the tumor interacts with the surrounding microenvironment. We're doing this utilizing a slightly different approach also when it comes to developing new small molecules. So we're working with a technology or a modus operandi, a way of working called fragment-based drug design. We'll come back a little bit as well on how that works and why we feel that that's the optimal way of actually doing new small molecules. The company is located in Stockholm. We're located in Huddinge, next to one of the Karolinska sites, where we today are a bit north of 30 employees. We are well labs and doing this kind of innovation work internally. So, all right, so kind of getting back and getting to the basics of how we do this and why we feel that this is the way to go forward to make this kind of molecules. When it comes to building a set of different kind of capabilities to be able to create this innovation engine. So what we built over the years is the capabilities within protein science, where we are able to isolate. So first and foremost, we figure out a good target molecule. And the question I always get is, well, how do you find all of these good targets? Well, it's a combination of many different things. One of them, of course, is that we have a very close understanding of the literature and where the scientific environment is moving. But we also, part of what we're doing is very close to basic research, where we kind of generate ideas internally in our labs. But anyhow, let's start with protein science, because we've identified a protein that we want to inhibit, and that one way or another is overexpressed in different kind of cancers. So what we do is we isolate it, and we can then make it in big enough volumes in our own labs to be able to characterize and look at it. What we're doing then, and for some of you that is following kind of how the pharmaceutical industry is working, there is a way of doing this. It's called high throughput screening. Essentially what you do is you build a huge library of so-called ready molecules. And that's normally anything from 500,000 molecules. Some companies have millions of molecules. For us, we're doing it differently because we see that even though these are ready, it's essentially a way of, when you do this and you screen these towards your targets, you're looking for something that's going to fit and have an effect. for us, it's more of a brute forcing the target. Think of it about the target as a lock, right? So you're looking for the key to essentially, unlock this. And by brute forcing it, essentially, it's like you remember the janitor's key ring, right? And you have all of the keys, and you're just trying everyone until you find something that works. We do this differently because we can make an image of the lock and ready and bespoke make the key to actually fit there. And that we can do by utilizing structural biology. So, and having and knowing that and seeing how that works, I'll come back to that in the next picture as well. When we have the understanding how our fragments that we start with, which is the starting point, when we can see how that fits into the binding pocket of the protein, we can utilize computational chemistry. And it's also a question I get, especially in this day of age, is like, how do you utilize? I mean, AI is coming, and how do you utilize that in your company? We've been working with that since the company was big data has been part of our modus operandi all of the time. And we actually have a dedicated set of skills called computational chemistry, which are chemists that are essentially working through their computers, being able to then make a prediction of how the best molecule, having the starting point, what would be the best molecule. What is also unique for us and not that common in the biotech is actually we have a fairly large set of medicinal chemists in our own chemistry lab. So that also means that we now have the blueprints of what we're going to build. And now that blueprint is handed over to the medicinal chemist that will actually make the molecule. And when we have a molecule, we can test it in our own cell labs. And this becomes an iterative step because continuously you learn something new when you do this, and there are ways of optimizing the molecule. But being able to build this from a small fragment and building it into a molecule, we can make sure that we also make very specific and very potent small molecules. So I'm going to try to visualize this a little bit for you. So what you see behind me here is a target protein. And the target protein has a binding pocket. They're intended at active sites where the protein utilizes its action in its body. And normally, I mean, there's of course a biological molecule that one way or another fits in there and kind of, amplifies the action of the protein. And having the benefit of actually being able to see this, we can make bespoke molecules that essentially works as a clone. That needs to be specific enough that it only acts on this protein and no other protein else in the body, because that's normally the case when we look at the history of making small molecules. It works in this binding pocket here, but it also happens that it also has some affinities to other proteins. That means you will have off-target effects. Sometimes there is tox, and sometimes it just adds on another effect. We want to make sure that we are working here and in this place only, and only in that kind of protein. All right, so that's a quick recap on fragment-based drug design. So the question is then, being a biotech and working exclusively only in preclinical, discovering preclinical phase, is there a business case to make, right? Everyone is talking about you need to make the clinical trials, you need to generate the clinical efficacy, and at least show some kind of science in patients. Well, the matter of fact is actually the market is showing something else. So this is the data from Q3, the accumulated data up until Q3 last year. So for the first 3 quarters of 25, where essentially 64% of all of the deals were made in the preclinical phase. And we still remain a healthy, good portion in oncology. We're seeing now some change, of course, an increase, alluding to the previous speaker in the cardiometabolic space, but it continues to be an area of high interest. And what we're hearing as well is there are a lot of companies increasingly looking at early assets because it's a more opportunistic approach. They understand that there will be failures along the way, and being able to tap into a broader set of innovations early is of interest, especially for companies that are not very close to a patent cliff, that has a balanced approach to the portfolio. All right, so how does that translate into sprints? Well, we made a fairly significant deal just before the holidays, where Gilead acquired wonderful compounds. Wonderful compounds, wonderful programs, because they essentially buy all of the innovation we made up until an early preclinical stage. For that, we got an upfront on signing on $14 million US, and the total deal value, including that 40 million, is $400 million US. We got some good traction globally as well, being featured not only in Sweden, the Nordics, but also on the other side of the pond, of course. Then maybe the feature was more Gilead by me, and in Sweden was more the sprint selling. But anyhow, I'm happy with that. For us, this is important in many ways. One, over the years, we've said to the investor community that the big pharmaceutical companies are looking into the innovation we're making. But it's, continues something we said. Now we can actually prove it. It's also a proof of the quality of the work we do. For those of you who don't know the way this works, when you sell these programs to the bigger pharmaceutical companies, you go through, a big due diligence. Essentially, they're looking into everything. I mean, everything. And that goes through the data package, looking at how the company works, how it's operated, and really, qualifying the companies, make sure that they're buying from a good source, and also that science helps. So for us now, it's not only the sense that we now are well funded, but it also means that we've been qualified. And I can see that also when I'm now out talking to potential partners, that the set of qualifying questions that's normally part of the introduction, the first 10 to 15 minutes, are no longer happening. Instead, they can see the made deal with Gilead, and that can qualify as support. But it's not the first deal we made. We've actually made 6 deals over the course of the last 16 years. And well, actually, over the course since 2015. We made a number of small partnerships also before that, when in the early stages of the company. So you can now see the biotech deal data from Sweden is primarily then from the listed companies, because that's where you have the full availability of the data. You can see some known names of Saniona, you might know, has made a couple of deals. Biortic, for those of you that follows the Swedish market, also made a couple. But out of these 28, that's been done over the last 20 years, we've done 5. And to be fair, if we have extended this a little bit more, I think our number 6 would be just. So where we're so we made a number of deals. I think we're also qualified ourselves now of actually being able to make deals. There's one of the benefits of having some age to the company. We built a good reputation among the industry, but also we built brand globally. We're well known and not only world famous in Sweden, but now also world famous a little bit more outside of that as well. So we're doing well. We're now seeing other ways of make sure that we're on the track of being a sustainable, profitable company. So of course we have over the years made this the classical license agreement. We received some milestones and contract research. We're now seeing that there's an opportunity to for people to actually acquire the pipeline programs out of the pipeline. We're also seeing an interest as well. Coming back to where I started, telling you about the early discovery preclinical, is essentially all of the programs we have in our pipeline. It's a good starting point as well for a new company. So we're seeing some increasing interest as well from venture cap and other sources of capital, where you're looking at our pipeline and seeing if this could be a good starting point for a company, and those discussions has been had. So far, we have not reached a conclusion on anything of that, but it's something we're openly and transparently telling to potential partners that we're willing to have those discussions. But it's also getting on the track and being a sustainably profitable company. We also need to make sure that we have starting to get a little bit more balance in our cash flow. And that also means that we've now built the lines. We have the capabilities. We also have some opportunity to expand in the current facilities we're sitting in. And that also means then that we're now looking to potentially adding on one or more platform collaborations. So it kind of gives us the opportunity for someone that might not see the targets they want on the list, but they do appreciate the way we're working, and to get that where they have the opportunity to come to the target that we can then work with together. All right, so I'll give you just a few highlights of the program we have that's remaining of the list of the tricks one. So we're working, as I said, primarily in the space of oncology. Bear with me, I will come to a slide again shortly when you will ask yourself, well, that's not oncology. I'll explain why. So DCPS, we're working with two targets here. This is something called synthetic lethality. Essentially, when you're looking at tumors, we're looking at tumor types where you have one dysfunctional gene and one functional gene. Normal tissue, you will have two functional genes, which means if you block out one of them, the other one will kind of cover and make sure that, you know, the loss of this protein is not functioning, but I will cover up that and make sure that this process works anyhow. Synthetic lethality is when you have one part of the protein is not there, it's not working, and it's either low or not there at all. That means if we are blocking out the other protein, that means that the function of the cell will disappear. And normally then it will die. And that's a way of treating cancer. It's a very smart way of doing that, because if you're normal tissue, both will be there, which means that even though we're blocking one of them in healthy tissue, there will be a backup plan in the cells to ensure that that continues to work properly. We have that in currently two programs. We have a DCPS program in AML, and we have our BRK1 program in glioblastoma, both of which we recently released press releases where we're showing the data. We've been able to publish some of the data on the DCPS program in a number of publications now, and we're currently, let's see what time it is. We might actually be right now presenting this in a poster session at AACR, which is one of the biggest cancer meetings in San Diego, taking place right now. And BRK1 is, as I said, we can go to glioblastoma, and we also published recently or issued a press release that we can now prove this in an animal model that it actually works for BRK2 low glioblastoma cells. We have EPS34, which is an autophagy inhibition. I'm not going to go into detail, but this is a super interesting program as well. We do have a drug candidate, and that's now being very mature towards GLP tox. And now getting to where I said that I might need to explain myself a little bit, because I said we were an oncology company. The thing is, when you are working in the immune oncology space, where one way or another we're going to modulate the immune system, and sometimes that also means that we are, in the borderline between oncology and immunology and inflammation. And this is one of these cases where we saw that we would like to explore or pivot part of the program. So we do have some really interesting data on the RLC, on the NNMT program in solid tumors. Essentially what it does, as I said, I talked about the microenvironment before. So this is a way to modulate fibroblasts around the tumor. The thing is that, and there's still really good data, and we're not abandoning the oncology track, but we also see some good recently published data on NNMT in chronic kidney disease. So we said, all right, the benefit of having a program like ours is that we have a solid chemistry foundation. That's going to remain no matter what the indication we're working with. We're now seeing an opportunity to also expand this in another disease area where there's a high unmet need and a high appetite for acquiring compounds. So we pivoted into chronic kidney disease. We showed we've got some really interesting antifibrotic data and shown that this could be a really good target in that disease area. And then we also have a collaboration, a program that has been part of the company for a while, where we see that there is a potential in the field of MASH, also in the antibiotic sense, where we have a very solid in vivo proof of concept where we show reduction in fibrosis and inflammation. So this is pipeline. The purpose of this company is to build as a broad pipeline as possible in this space. It's a cost-efficient way of building a number of assets, utilizing the model we have. And we are then having currently these 5 programs in the pipeline, but we also have a number of undisclosed programs. And we are continuously, it is an innovation engine, we are continuously looking at new interesting targets. So, Spread Bioscience is our investment. I mean, we have a unique platform that can prove in itself. We've been able to do a number of deals, and I would say that the validation of the Gilead deal has, for us, it's definitely a catalyzing effect for being able to reach the vision we have. We do have an attractive and diversified portfolio. We also have the opportunity within the facilities and the setup we have to scale it. We have a strong pipeline of programs, and we have existing and, of course, potential new partnerships. With that, I'll open up for questions. Thank you very much.
Any questions?
Maybe short blocks.
How does the business model scale when you sell the programs this early?
So how to scale? So one of the benefits, of course, first and foremost, is a very cost-efficient machine. So it is in the stages where we're not making huge investments in going into kind of the IND-enabling talks, or then eventually going into the clinic. And the whole purpose of this business model is to make sure that we're not a binary company. With that, I mean that we should not stand and fall with a single data. So for us, this is an opportunity for us to continue to diversify our portfolio and to continue to build out. And also, coming back a little bit to what I said before, we have built a good brand, we have a good understanding, we have a good network, and the more things we have in the portfolio, the bigger opportunity is, of course, that we find the right partner and continue to get people to partner with us.
I think it's many of your programs, they are very premature. Before preclinical phase. I'm surprised that Gilead would pay so much for a program in that early stage.
First and
Is that
foremost,
normal?
so we see that there is a number of deals being made in preclinical stages. The way we see this, we have the Danish case of AbbVie and Gubra that is, we're not playing in the same, on the same ballpark here. But we see that there is a good appetite to acquire good innovation. I have to remember that everything we do is first in class, or the intention to be first in class. But we're also, with what we're doing, solving some problems, especially in I wouldn't call it undruggable, because apparently we can find a solution for it, but maybe as historically has been seen as undruggable, or really hard to make good selective potent inhibitors for. And when we do that, they are willing to pay.
That's very good. Any questions?
Oh, Fleming Peterson, yeah. Could you tell a little bit about the company accountant numbers, balance sheets, and so on?
So I can comment on the recently released numbers. So we received, as I said, a little bit north of 130 million Swedish kroners. So $14 million just before, well, in November, essentially. And we currently normally on a normal month have a burn rate around 5 million Swedish kronor. So it kind of gives you an understanding of approximately where we are at. I won't comment on the you have to wait on the outcome of the first quarter until May.
Okay, but still 5 million.
Around, yeah.
Month.
That's
Money for some time.
And bearing in mind that's with a little bit more than 30 employees in our own labs. So
My name is Alexander. Do you expect a deal within this year?
I've said probably before, and also in a written comment, that's why we're working for it. Yes, we see that we have an opportunity. We have this is probably the most mature pipeline the companies have ever had, and together with the validation of the deal with Gilead, of course, that's what we're working towards. Yes.
If a drug is really promising, isn't it tempting to take it all the way to scale it more than just sell it off in that early stage?
I do understand it. I mean, coming back to my history and what we're working before, I spent most of my career in the early phase of transitioning compounds from preclinical into clinical. It's the most amazing thing you can do because it gives you tangible clinical data and keeping on understanding how this is going to work. For us, we say no. We're focusing on what we're great at. And we're great at discovery and early preclinical. And the benefit of making sure that we do that is also that I can reinvest in the personnel I have. So I'm not going from, I have a set of skills in the early part, we're moving it towards the R&D enabling studies, getting into clinic. And now we need a different kind of, we need more money. That means we also need to make some hard decision. It might be that we say thank you and goodbye to the people we had in the beginning. We're now entering into interface. I see the benefit of reinvesting that and continuing to build great early innovation that someone is willing to pay for. In the future, who knows? I mean, if we have the financial stability to be able to press things further, we might. But here and now and for the foreseeable future, we will continue to be focusing on what we're great at.
You showed us that you have made, I think, was 5 deals.
Okay.
Okay. Was that in the size as the one with Gilead or
No.
bigger, smaller?
No. I think if I go back to that slide, it is in increasing order. And that's a benefit. And it also gives some expectation on my end, and also the expectation on other people on us. But it's been increasing over the years. But upfront, that's not, I think the biggest upfront has been $3 million. But that has been a different setup on the deals. That has been a licensing setup, where there's been an active license on our know-how and IP.
Well, you confirmed that you're intending to make a deal this year as well. Is that a practice that you want to make one deal each year? Or?
I like to make as many deals as I can every year, but at least one. Yeah, I mean, if we're going to be focusing, what we're trying to build now is a portfolio of a number of active collaborations, which means that we're building a portfolio of potential future milestones and royalties. And so that's going to be the focus. I don't need to make a deal today, and that's actually the best negotiation position you can be in. At the moment, we actually have money in the bank to be able to say no.
But we have to wait to see.
I will make sure you will know when it's done.
In due time.
In due time.
Well, thank you very much, Johan. Thank you very much for coming.